recording. Okay, I'll begin then. Hi, I'm Madison Juliana Oliva. I'm also a math major, and I'm going to be talking about my research on my agent-based model of neutrophil chemotaxis during the wound healing process. So this is a bit similar to Ryan's, just a different aspect of wound healing. So you're familiar with the wound healing process. You get a cut in your skin, you put a Band-Aid over it, and then later you're left with a fibrotic scar. Underneath this all, you get a bunch of different mechanisms, one of which is the inflammatory response. In this process, it begins with the calcium waves, hydrogen peroxides, DAMPs, and other signaling pathways coming in and actually bringing in inflammatory cells to fight infection at the wound. I will be talking about a specific inflammatory cell called the neutrophil. So the neutrophils are regarded as the body's first responder. They are short-lived white blood cells that fight infection and disease, and they're recruited within the first few hours of injury. They develop in the bone marrow, and then they patrol the bloodstream on the lookout for where to fight infection and disease whenever a wound appears, or just throughout the body. We know a little bit about neutrophils over the past couple of decades, but it wasn't until 2008 when we were able to actually visualize what the neutrophil movement was at the wound space. Before that, experimental limitations didn't allow us to get clear images of this actual movement. But two intravital imaging studies at the time in mice revealed neutrophil behavior at the infection sites. What was revealed was a phenomenon among neutrophils called neutrophil swarming. Researchers have been able to report more and more about this phenomenon to put together this 5-step attraction model of extravascular neutrophil swarming. Basically, when you get a wound, the neutrophils will respond in these 5 steps. The first is swarm initiation, and then the swarm will amplify with the next 2 steps through the first secondary cell death of neutrophils, and then more signals relaying through the intracellular signal relay. And then in step 4, the swarm will grow even more to aggregate on each other, and the tissue will begin remodeling for the wound. Afterwards, in step 5, myeloid cells will come and be recruited, and the swarm will resolve itself and start breaking away and basically just stopping, so we don't have overswarming. This swarming process is really important because it's a very necessary part of the wound healing process. Neutrophil swells in their swarm will basically eat a bunch of infection and disease that would otherwise harm your wound even more. So having this swarm actually effectively come into play and then not overswarm is really important to keep your body safe. Because it's a more recent discovery, there's a gap of mathematical models of this process. Mathematical models offer insight to cellular behavior, and this is necessary to develop treatments for any wound-related conditions people might have. In this case, if a neutrophil swarm was to get out of hand, either by overswarming or underswarming, the host is at risk for either the swarm attacking the actual parts of your body that you don't want it to kill, other than infection and disease, or it can just not even come to play at all, and infection and disease will run rampant in your body. So once we actually better understand the cellular behavior, we can create substitutes, cell-based therapies, and other actual treatments to take care of these conditions. That leads me to the goal of this research. We want to develop a hybrid agent-based model on neutrophil swarming dynamics to identify the key factors guaranteeing neutrophil swarm success. Once we know this, it can help in the development of different treatments just so we have a better understanding of what to affect to make the neutrophil swarm succeed. We do this in a variety of different steps to get to our final model, but the first is to model the signaling dynamics of our actual wounds and how it diffuses in the space around the wound region. So to do this, I calculated this reaction-diffusion equation as seen here. And I'll explain this in a little bit, but the main idea is at each point on the wound space, a different amount of signal will be produced that will come in, and the neutrophils in the area will detect it and then start getting attracted and swarming onto the wound region, like in real life. So to explain this equation a little bit, it's basically another ordinary differential equation, and it calculates at any given point the amount of signal. So this would be the resulting signal at a given point at a given time. And it depends on whether there's a wound point at that area. So basically, is this part of the wound or not? And it also depends on the points around it. If those have signals, it'll diffuse into this current signal space. We solve this using finite difference methods with also forward Euler, and we develop this in C++, meaning we're going to calculate all of this in C++ for a faster speed, and later I will bring this data into MATLAB and visualize it. On the right, you can see a few stills of the resulting chemotaxis model, but I will be showing a video on the next slide actually depicting this. So from that signal dynamics, we come up with this chemotaxis model. Here, I'll show a video of the model with the signals diffusing from the wound area from the center. The neutrophils aren't pictured here. We're just trying to show how the signals from the wound actually move. So let me play it for you. Here you can see the colors change as the signal strength increases from the center and diffuses out towards the rest of the space. So afterwards, the next step is to model the neutrophil movement with an agent-based model, also using C++. An agent-based model is a type of mathematical model where you focus on your agent, which in our case is the neutrophil. You determine a bunch of rules of movement and stuff around the space that will affect how a neutrophil agent will move. And you'll just play it and see how the neutrophils will go around. And you can determine a basis of cellular movement behavior as a result. So our rules of movement for our neutrophil agent is first that it would move on lattice, meaning that it's stuck on a grid and will only move in 4 directions, up, down, left, and right. The neutrophil cells will follow the rules of movement determined by the wound signal strength at a given point. So if at its current point, the signal strength is less than the potential point it will take, it'll move towards the other one, just because this will in turn cause a bunch of swarming towards larger signal strengths towards the wound. And that will be determined by the chemotaxis model I showed before. Based on the calculations of that one, it will determine whether the neutrophil cell will move in this agent-based model. And then finally, the neutrophil cells will have a lifetime counter attached to each agent. And once it reaches 0, it will leave the system reflecting cell deaths. And with all that data, we're going to take it and visualize the model with MATLAB. So here is just the agent-based model by itself. It's not depending on the signals I calculated, just to show you how the actual movement is. As you can see, they all start swarming towards the center in that wound region, and they disappear over time to emulate cell deaths. At this point in research, we were able to combine both the chemotaxis and the agent-based model to get the resulting hybrid agent-based model. On the left side, you're going to see the chemotaxis part, and on the right side, you're going to see the agent-based model with the neutrophil cells moving around. They're both the same area. It's just one graph shows more about the signals diffusing and one shows the neutrophil cells, so it isn't too confusing on the same graph. So basically, in this stage, we currently have completed step 1, the swarm initiation, and are well on the way to finishing step 2, which is the first swarm amplification through secondary cell death. Now that the cells can die, we just have to bring in more neutrophil cells from the surrounding areas. With that in mind, I have some next steps planned for this work. So the first one is to update the agent-based model to account for new neutrophil cells entering the system. Currently, the only ones in the system are ones that start there, but that isn't true to real life where more cells will come in from farther away. And then next, I would like to update the equation to account for present neutrophils. Right now, the actual signals will continuously increase at a given point, but it doesn't take into account neutrophil density that will take up current actual signals. And then next, I just want to complete steps 3 through 5 of the rest of the swarm. Here are my references, and I'd like to thank you for listening and also thank my PI, Dr. Wang from the Department of Mathematics. Yes, let me stop sharing. Okay.
And you can stop recording too.
Oh,
Madison.
yes.